So we move on now to talk about what happens once you actually get those fatty acids into your cells. And so again, we're going to focus in this lecture on beta oxidation of fatty acids and then fatty acid synthesis from acetyl-CoA. So again, this is what we can think of as a large substrate cycle depending on whether we're in a need state and undergoing beta oxidation or a fed state and undergoing fatty acid synthesis. So again, conceptually, we'll need to think about how we would put ourselves in a need state. And again, thinking about whether we're in a global or a local need state. And then a mantra that we'll sort of talk about with beta oxidation is oxidation, hydration, oxidation, cleavage. And what we'll see is we actually reverse that mantra when we think about fatty acid synthesis. So in that case, it's going to be reduction, dehydration, reduction, and then condensation. And we'll actually think about that in the reverse direction as well. We'll do condensation first, reduction, dehydration, reduction. So a little bit in terms of thinking about how we actually get fatty acids into the destination where beta oxidation, or into the location where beta oxidation occurs, which is the mitochondria. And then again, we'll talk about the process of beta oxidation. Okay, so the first thing that we need to think about, remember, all we've done is get some fatty acids into the cytosol. So here we have a fatty acid that's gotten delivered to the cytosol, come in from the bloodstream, and now what are we going to do with it? Well, remember that fatty acids pretty much have a pretty stinky functional group, right? They just have a carboxylate there. We really can't do anything with it. So the first thing that we're going to need to do is activate these so we can actually start doing some chemistry. So activation is almost always going to require a little bit of money input. So that's why we see ATP here. We're going to see ATP is going to facilitate this process by generating a good leaving group. So now we're going to have our fatty acid that has gotten activated by putting an OAMP group on there. Remember, we've seen that before in our reaction mechanisms. So once we put a good leaving group on there, then we can have a weaker nucleophile like CoA with its thiol group here, nucleophilically attack that carbonyl on that fatty acid, displacing our good leaving group and generating a fatty acyl-CoA. So the first step is we need to take this fatty acid that had no functional group, no ability to really do much of anything else, and we convert it into a fatty acyl-CoA so that we can start doing some chemistry. Now the next piece is actually taking that fatty acyl-CoA and getting it into the mitochondria. Because remember, right now what we have is this fatty acid that's attached onto a CoA, but it's sitting out in the cytosol. We need to get it into the mitochondrion because that's where chemistry occurs. So again, we can see we've got cytosol here, we've got the matrix, so we're going all the way on the innermost compartment of the mitochondria, so we have to get past the inner mitochondrial membrane, and here's the transporter that we need to get through. It's called a carnitine carrier protein. And what it's allowing any protein to do is if you wear this carnitine hat, if you will, you can get in. So here's what the carnitine hat looks like, and notice that it's got an alcohol group that we have here. Well, remember, a trans-esterification reaction would basically take one ester. So if we've got an ester group here, and actually let me draw this out here. If we've got an ester group here, and we've got one OR group, and we want to take another OR group and swap it out for that, we can take and swap out this alcohol essentially for that guy coming off as an alcohol, and that's called trans-esterification reaction. Well, if one of these guys happens to be a sulfur group, meaning that it's a thioester, we can still do the same chemistry, but what we're going to do is we're going to swap out the guy that was the alcohol here for the guy that was the thiol here. So we call this trans-thioesterification, and really this carnitine group is the hat that I'm talking about. So what we do is we take our fatty acyl-CoA and we get it to put on this hat. Once you have this hat on, you can pass through this carrier protein and get into the mitochondrial matrix. Then you just reverse that chemistry, take your hat back off, and now you are a fatty acyl-CoA that lives in the mitochondrial matrix. Once we're in the mitochondrial matrix, that's when we can start doing our beta oxidation chemistry. So again, this is, just summarizing that as part of one of your homework questions. We take our fatty acid, we need to have some money, and then CoA come in. We're going to generate a fatty acyl-CoA there. We get fatty acyl-CoA to put on its carnitine hat so that it can pass through this carnitine carrier protein and become a carnitine hat-wearing fatty acid in the mitochondria. We do another trans-thioesterification reaction to take that hat off. And now we've got a fatty acyl-CoA that's in our mitochondria that is basically now ready to undergo beta oxidation. All right, so here is in a nutshell what happens in beta oxidation. There's lots of enzymes, there's lots of really neat chemistry here, but we're not doing what we did for glycolysis and the citric acid cycle here with fatty acid oxidation. I'm not having you guys know structures or enzyme names or anything like that. I just kind of want you to know the nuts and bolts of what happens. So this is actually a very simple picture. Each of these kind of orange spheres represents a carbon atom that we have here. And so we've got our fatty acid, and here we've got to cut the money end of the molecule, which is where our functional groups are. So here's the thioester. Here we've got our CoA group there. So again, we're going to call that the business end of our molecule because that's where the functional groups are. So 4 steps to this chemistry here: oxidation, hydration, oxidation, cleavage. So again, we're going to look at the functional groups that we have here. So if we're looking at those 2 carbon atoms, it's basically a methylene group that we have there. Okay. First step is we're going to do oxidation of that alkane to an alkene. Okay. Oxidation of alkanes to alkenes was stuff that we saw in the citric acid cycle, right? We took succinate that sucked at having functional groups, and we used FADH2 to generate fumarate. So just a little bit of a review there of stuff that we did need to know in terms of names. We don't need to know any names of anything here, but I want to highlight the functional group changes that are going on. So alkane to alkene, that kind of oxidation chemistry is going to see FAD to FADH2. Next step is we're going to hydrate our alkene and we're going to make an alcohol. So again, alkene to alcohol was what we saw when we converted fumarate to malate. So water is an important cofactor that we need to have there, and we generate an alcohol. Now the last step here is we've got oxidation of our alcohol group to a ketone. So again, we've got oxidation of our alcohol group to a ketone there. That type of oxidation chemistry we see with NAD+ being reduced to NADH. At the end of this point, we have a beta keto thioester. That's a really important thing because anytime we've got a beta keto carbonyl containing compound, the bond between that alpha and that beta carbon is what we call labile or easily broken. We're going to look at this mechanistically on the next slide here. But basically, we're going to be breaking and doing carbon-carbon bond cleavage chemistry here. What we're going to look at to sort of see that chemistry is thinking about reversing the chemistry that we see with citrate synthase. So remember, if we take citrate was made from oxaloacetate and acetyl-CoA. Again, if we reverse that process and we want to break those apart, we're going to be thinking about that as being reverse citrate synthase chemistry. And maybe you remember from our citric acid cycle chapter, condensing oxaloacetate and acetyl-CoA was what we call a Claisen condensation. So this is actually going to be a reverse Claisen condensation to do this cleavage chemistry. So again, I'm not going to go through the mechanism for this. There's another video that kind of highlights this mechanism. But again, here's our beta keto thioester. This is the bond that's going to be labile, so that ends up being broken, and then we end up at the end of the day also hydrolyzing off that, that thioester group. All right, that's all I'm going to really say about the beta oxidation process. I do just want you to remember those general steps. Talking here a little bit about ketone bodies. We addressed these already in chapter 9 back in Biochem 1, but I do want you to know that all ketone bodies are ultimately made from acetyl-CoA. And so if we look at the structure of these 3 ketone bodies, I do want you to know how they're related. And I want you to think about acetoacetate as kind of being the central guy here, because this is related to beta-hydroxybutyrate via redox chemistry. If we reduce this carbonyl and generate our alcohol, that's how we get beta-hydroxybutyrate. And again, oxidation of that alcohol back to our carbonyl, our ketone there, is going to generate acetoacetate. So these are just two different flavors of essentially the same molecule that are interchangeable by redox chemistry. But acetoacetate and acetone are related in a different way. Remember, we've got a beta keto acid here, so this carbonyl or this carboxylate group will easily come off as CO2, or relatively easily, because we can generate a resonance stabilized enolate intermediate. And so beta keto acids easily decarboxylate, and in this case, we generate acetone. So this pathway is really important here, because if you start building up excess acetoacetate, one of the ways that your body can get rid of it is undergo this decarboxylation chemistry and generate acetone, which is much more volatile, and you can actually exhale that. So as you elevate acetone levels in your blood, you can actually put it into and volatilize it in your lungs and exhale it out. And so that's a way of getting rid of excess ketone bodies. So we've talked about that before with people who are diabetic. Oftentimes you might smell a sweetness on their breath, or if they're in this state of ketoacidosis, and that's because they've got excess levels of ketone bodies in their blood, and their body is desperately trying to kind of rid itself of that by breathing out the acetone. Again, just a homework question here that we didn't have necessarily room on a slide for. We've already sort of talked about this a little bit, but just to summarize it again, the importance of acetyl-CoA as a branch point metabolite. We've already learned about carbohydrates. We've now learned about fatty acids being able to synthesize acetyl-CoA. We'll learn the metabolic pathways in our next learning unit for how amino acids can make acetyl-CoA, but you've known for a long time that all three of our main biological macromolecules can be converted into acetyl-CoA. So what happens to acetyl-CoA ultimately depends on whether you're in a need state or a fed state. Again, if you're in a need state, you're going to be locally, if you're again in the liver, breaking it down via the citric acid cycle to generate the ATP and waste products that allow you to generate the energy that you need. However, if you're helping in a global sense, your liver will take acetyl-CoA, convert it into ketone bodies, which it then exports for the rest of the body to use. Now again, acetyl-CoA, if it's in excess, and this is where we're moving on to next, your liver is capable of taking excess acetyl-CoA and undergoing fatty acid synthesis to make it into fatty acids, which then can ultimately be assimilated with glycerol to make triglycerides and offloaded for storage in adipose tissue. All right, so before we move on to sort of our understanding of the synthetic pathway for fatty acids, just wanted to take, this is a nice slide to sort of summarize and highlight the differences that we see between beta oxidation and then fatty acid biosynthesis. So a very important distinction, and this is going to be a critical component to how these processes are regulated, is their cellular location. Fatty acid degradation via beta oxidation occurs in the mitochondrion. Fatty acid synthesis, when you have excess acetyl-CoA, occurs in the cytoplasm. So it's very important that we have a robust kind of delineation between these two processes, because we do not want to have a futile substrate cycle where we're synthesizing fatty acids in the cytosol and then we're just breaking them down in the mitochondria. This would be a massive futile cycle if it was allowed to just kind of occur like that. So we've got really tight regulation of this, and we'll talk in our next lecture about how that happens. But highlighting again what happens here in our process of beta oxidation. So we're going to have oxidation, hydration, oxidation, cleavage. And we see here these oxidation chemistries occur with the generation of the reduced cofactors NADH and FADH2. So we essentially reverse that process with fatty acid synthesis. So the 30,000-foot view again of fatty acid synthesis is condensation, reduction, dehydration, reduction. And so one of the things that's key too is the cofactors that are required for this reductive biosynthesis in both steps are going to be NADPH. And we talked about where we get NADPH, right? We get NADPH from the pentose phosphate pathway. That's how we synthesize that. All right. So homework question 13 and 14 kind of deal with addressing how we get fatty acids destined for storage. If we've got excess fuel, how we get them where they need to go. So remember, if we have any excess acetyl-CoA, whether it's made from beta oxidation, whether it's made from pyruvate dehydrogenase chemistry, it is going to live in the mitochondrion. So how do we get it out to be acetyl-CoA in the cytosol? Well, that again is one of these transport issues. We do not have a transporter for acetyl-CoA. It's kind of critical that we often don't see transporters for crucial branch point metabolites because we really want to control where they're going to be. And so what we actually do is we tap into a citric acid cycle enzyme. We tap into citrate synthase, but it's really important at this point that citrate synthase kind of disassociates from being part of the citric acid cycle because we don't want to funnel it through to make citric acid cycle products. We actually want to be able to siphon off the citrate. So again, we're going to use citrate synthase. We're going to use some of the oxaloacetate that's present in the mitochondrion. And again, it's all right to use some of that because if we're not in a need state, we don't need to be doing citric acid cycle chemistry. So we're going to make citrate that's in the mitochondrion. We do have a citrate transporter. So this tricarboxylate transport system will transport citrate. So in essence, we disguise acetyl-CoA as citrate by coupling it with oxaloacetate. We transport it across that inner mitochondrial membrane, and now we've got citrate in the cytosol. We do essentially reverse citrate synthase chemistry, but we don't have citrate synthase that lives out in the cytosol. Remember, we talked about this when we talked about mitochondrial and cytosolic isoforms of malate dehydrogenase, but we don't have the same with citrate synthase. Citrate synthase only lives in the mitochondrion. When we get out to the cytosol, we do have an enzyme that does the chemistry to reverse citrate synthase, but it's called citrate lyase. What we get then is we get back our acetyl-CoA, but now it lives in the cytosol. And now we've got oxaloacetate that lives in the cytosol, but we can again then use the malate aspartate shuttle as a way to get oxaloacetate that's in the cytosol returned to the mitochondrion. So we're not really depleting that mitochondrial pool of oxaloacetate. So again, homework question 14 here really deals with the mechanism here for citrate synthase. So when we talked about beta oxidation chemistry, we were sort of talking about that reverse Claisen condensation. Now we are actually talking about that Claisen condensation chemistry. So we're going to get to that in just a minute here. All right, so now we have excess acetyl-CoA that is waiting out in the cytosol for us to do something with it. It got out there because we siphoned off citrate from inside the mitochondrion, and now we've reassembled that citrate back into acetyl-CoA that's sitting in the mitochondrion. Okay, so now we've got acetyl-CoA that's sitting out there. So one of the things I want to highlight is we need to do a little bit more manipulation in order to make acetyl-CoA ready to do some chemistry. So we're going to call this sort of tagging and then activation. So the first thing that has to happen is we've got acetyl-CoA on CoA. So look at, and again, you don't need to know the structure of this, but this is what the structure of CoA looks like. It's derived from a nucleotide, so it's a nucleotide derivative. So we've got sort of a nucleotide component here. We've got this long arm, and then importantly, we've got a thiol functional group. So that's why we often write CoA as CoASH, reminding us that it's got that thiol functional group at the end. Okay, so that is where our acetyl group is attached right now, onto CoA. We need to get it onto acyl carrier protein, okay? Because acyl carrier protein is one of the sort of machine shops, if you will, in our giant fatty acid synthase factory. Okay, so we need to actually do one of those kind of trans thioesterification reactions to get our acetyl group to be on ACP, on acyl carrier protein. So what we're going to see is we're going to have to put some of our acetyl-CoA onto acyl carrier protein, and then some of it needs to actually be activated to do chemistry. Okay, so that's sort of the second part. So the first part, the tagging part, really has to do with taking our acetyl group off of CoA and loading it onto the factory that's going to be doing the chemistry. So that's sort of the tagging part. Activation is, you know what? Here's acetyl-CoA. Acetyl-CoA is not going to spontaneously have this carbon atom be nucleophilic. So what we need to do is a little bit of magic in order to make it be nucleophilic. Okay? And the magic happens with this important enzyme. This is the money enzyme for fat synthesis. It's called ACC, which stands for acetyl-CoA carboxylase. So what we're going to do is we're going to take acetyl-CoA and we're going to put a CO2 group on. We're going to put a CO2 group on that carbon atom. Okay, now that's not easy chemistry to do. Remember, we've seen chemistry like this before. Pyruvate carboxylase did this chemistry. That's why we've got this written down here that this is a homolog. Remember, what we needed to have for that is we needed to have vitamin B7, so we're going to need to see biotin here. So biotin, remember, is our carbon dioxide kind of transfer guy. But again, that's not without a toll to pay. That's not without a cost. So we've got bicarbonate here is going to be the source of the carbon, but we need to pay a toll. So we need to have some ATP involved with that. So this whole activation process is basically AcetylCoA carboxylase, taking acetyl-CoA, the required vitamin B7, and then bicarbonate and ATP for money, and generating what we call malonyl-CoA. Okay, so why is this important? Okay, so this is kind of the reason why we do this chemistry. Here's acetyl-CoA. We want this methyl group to be a nucleophile. Okay, you know, it's not easy to do that. We're going to see when we look at a couple of mechanisms here, there's a couple of ways that can happen. But one of the ways that we biochemically can make that happen is we add CO2 to remove CO2. Okay, so here's the ACC chemistry here. We're going to use bicarbonate as our carbon source, ATP as our money, and we make malonyl-CoA. So we've added CO2 to the carbon that we want to be a nucleophile. Now, this is a beta keto acid, right? Here's our acid functional group. At the beta position is a carbonyl, okay? So that allows us to easily decarboxylate this. Decarboxylation of malonyl-CoA generates this enolate intermediate. So again, we wanted this carbon atom to be nucleophilic. Once we add the CO2 and then remove it, it is a nucleophile, which is awesome. That's what we wanted. So this is a theme that we see. When we want carbon atoms to be nucleophilic, sometimes what we'll do is we'll do this magic trick of adding CO2 and removing CO2, and that generates the enolate that we need to be a nucleophile. Okay, so a couple of mechanisms here that, again, I'm not going to go through the details of because there's a separate video that kind of highlights this, but two sort of different mechanisms here highlight kind of different ways that we can make carbon atoms be nucleophilic, okay? So here again is oxaloacetate and acetyl-CoA. So this is the chemistry for citrate synthase. And what I'm going to highlight here, these two mechanisms are different, but they're both ways that take acetyl-CoA and allow it to be nucleophilic, okay? So in citrate synthase chemistry, we actually deprotonate. We have in the enzyme active site an amino acid residue that can deprotonate that methyl group and make it be nucleophilic. So that's kind of like an ideal situation, right? You just made the nucleophile directly, you couple them together, and then again, that's how we generated citrate, okay? This is different than what we're doing here. In this case, we made malonyl-CoA. So here's sort of our acetyl-CoA portion that had the extra CO2 that we added. When we decarboxylate, that makes that guy nucleophilic, so that's how we make our nucleophile. It's actually going to react with another acetyl-CoA group, but how we made one of the acetyl-CoAs be able to be a nucleophile is we did our magic trick of adding and then removing CO2, okay? And again, we talked about this as not being a true mechanism. It's drawing mechanistically, just kind of showing nucleophilic attack, tetrahedral intermediate, decomposition, and then departure of our good leaving group. So that's how we do that carbon-carbon bond forming chemistry in fatty acid synthesis. Okay, so just going to highlight right here one other thing that we're going to get to the details of in just a minute. When we do this synthesis in our fatty acid synthase factory, we're going to have the malonyl portion is attached to the acyl carrier protein, but the acetyl-CoA is going to be transferred to a different kind of floor in the factory, if you will. It's going to be put on a different part of our machine. So that's why we have these two guys listed here. All right, so I'm going to flip through these slides because they were just when we were kind of explaining how citrate synthase works. And again, here were the mechanisms that I actually went through with the video. So that's what those slides were. And this, I think, was just looking at kind of comparing that to the chemistry that we see with pyruvate carboxylase. All right, so here is the chemistry that then happens with fatty acid synthase, right? So remember we had oxidation, hydration, oxidation, cleavage for beta oxidation. Now we're going to see the opposite: condensation, reduction, dehydration, reduction. Okay, so spent a lot of time on this slide, but again, what we're going to see is the reversal of sort of these functional groups. So we take a carbonyl, we take a ketone, we reduce it to an alcohol, we then dehydrate it to an alkene, and then we reduce it to an alkane. Both of those reduction steps are going to occur with NADPH. And then again, we're going to see elimination of water in our dehydration step. And then again, that condensation step was the chemistry that we've just been talking about. All right, so a lot on this slide, but let's kind of highlight sort of what's happening here. So again, we're going to have one acetyl-CoA group that got activated because we put it on our acyl carrier protein. Remember, we said we can't have stuff on acetyl-CoA. It has to be on the acyl carrier protein. So this is sort of our loading step, if you will. So we've loaded acetyl-CoA onto acyl carrier protein, and then we're going to load that malonyl-CoA, which was made by that ACC enzyme. We load that onto acyl carrier protein. So again, envision this big factory that we have, and we've got all these different domains that are going to be doing chemistry. We've now taken acetyl-CoA and malonyl-CoA, and we've loaded them on acyl carrier protein. Now what's going to happen is we're going to transfer the acetyl group, not the malonyl group, the 2-carbon acetyl group, to that Ks. So one of the things I want you to remember is that the acetyl group, which is going to be our electrophile, is going to be on Ks. We've got our nucleophile, and our malonyl is going to be on the acyl carrier protein. Okay? Now what happens is our machinery allows these two to come together, and we do our condensation chemistry. So we did our condensation chemistry, we made that new bond right there, and we coupled the 2 carbons from our acetyl group with the 2 carbons from our decarboxylated malonyl group. So now we've got 4 carbons from 2 acetyl-CoAs coming together, and then we've got to do the reduction, dehydration, reduction chemistry to get to have our full hydrocarbon there. And then essentially what we're going to do is we're going to reverse this process. What's going to happen is this guy, who we want to be an electrophile again, gets reloaded on the Ks domain. And we're going to have another malonyl group ready to act as a nucleophile loaded on another acyl carrier protein domain. This cycle is going to repeat until we get to have 16 carbon atoms that are totally hooked together and are fully saturated. Now, a homework question here asks, and there's lots of neat biochemistry that talks about this, but we're not going into any of those details. If you want to make it longer, no problem, just use elongases. If you want to make it unsaturated, no worries, just use desaturases. And at the end of the day, now we've made our fatty acids. But if we want to do anything else with this, we need to actually attach them onto that glycerol backbone. So here's a little bit of biochemistry with how that happens. Glycerol, remember, is a 3-carbon piece, so we actually derive that from dihydroxyacetone phosphate that we can siphon off of glycolysis. So we've got DHAP that we do a little reductive chemistry to get glycerol 3-phosphate. Now we've got a backbone here. If you add just 2 fatty acids to it, that's going to be helpful if you're making phospholipid bilayers, because that'll make your phospholipids. But if you're going to be storing these triglycerides in adipose tissue, you're going to put 3 fatty acids on that glycerol backbone and make your triglycerides. All right, so this was, again, a slide that we had that kind of showed us this machine, showed us this factory. And again, I want you to know what happens in different steps, which you could get pretty easily, I think, from this slide. But this is just kind of a nice summary slide that kind of highlights the chemistry that happens, the functional group changes that happen with each of these reduction or dehydration steps. Again, you do not need to know any names for these guys, but I do want you to know that we're going to have acetyl-CoA, or whatever our electrophile is going to be attached to the Ks domain. And then our malonyl-CoA. Malonyl-CoA is going to be always our nucleophile. That's going to be attached to the acyl carrier protein. And notice this acyl carrier protein has this really long arm, which means it can dip over into these different domains depending on the kind of chemistry that needs to be happening. So it's going to dip over here first to the KS domain, do chemistry with that electrophile, add those carbon atoms, make that carbon-carbon bond. And then we've got all of these functional group changes that have to happen, and so it dips into different functional group pools depending on what kind of chemistry should have to happen. I shouldn't point to the dimerization domain. That's not where we do chemistry. This is actually a factory that exists in two parts. So we actually have, that's where those two parts kind of like hooked together. Okay, so I'm not going to go through too much on this slide because this was just a way to summarize a lot of the same stuff that we talked about on this slide. All right, so at this point, we've kind of finished up the nuts and bolts of how we think about fatty acid breakdown. And again, fatty acid breakdown occurs in the mitochondrion, and it's going to generate acetyl-CoA. And then we talked about, well, what if you have excess acetyl-CoA? What if you've had excess fuel and you're in a fed state? We talked about how we get that excess acetyl-CoA via citrate out into the cytosol and then use that fatty acid synthase machine in order to synthesize fatty acids to be able to store them as triglycerides.